Hello dear students, I hope you are all fine in this period of pandemic, that is COVID-19 pandemic. Now in our this going on lecture series we are going to start with our new chapter or a new lecture series, that is of the chapter that is cell, the unit of life. We are going to talk about cell, which is known as the unit of life. So when you look around, you see both living and non-living things. You must have wondered and asked yourself that what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has. So what there is absent in non-living and what is there present in case of living, that can be our question in our mind. So the answer to this is the presence of a basic unit of life, that is the cell in all living organism. So this cell is the basic unit of life. Cell is the basic unit of life. We know that all organisms are composed of cells. Some are composed of single cell and they are called as unicellular organism, while others which are composed of many cells, they are known as multicellular organism. So first we try to understand what is cell. We are trying to understand and trying to answer the question that what is a cell. So if we focus on this question, then we get answer that when we look at a unicellular organism, these are capable of independent existence. They are of independent existence and performing essential functions of life. Performing essential functions of life. So this unicellular organism capable, they are live independently and they perform all the functions. So anything that is less than a complete structure of a cell does not ensure a independent living. Hence, cell is the fundamental structural and functional unit of all living organism. Now if we talk about the cell, Antony von Leeuwenhoek, if we talk about the discovery, first saw and described the cells. Later on, Robert Brown discovered nucleus. And the invention of microscope and the improvement leading to the electron microscope revealed all the So this electron microscopy revealed all the structural details of a cell. Now, so we have understood that cell is without a particular complete structure of a cell, the living is not possible. So next we come to the topic that is known as cell theory. cell theory combinedly given by a Sladen and Swan. But we first focus about a contribution of Matthias Sladen in 1838. He was a German botanist. We remember he was a German botanist, and he examined large number of plants he observed a large number of plants and observed that all plants are composed of different kind of cells. Different kind of cells which form tissues of the plant. At about the same time, another scientist in 1839 Theodor Swan who was a British zoologist studied different type of animal cell and reported that a thin outer layer which is known as plasma membrane. He reported a thin outer layer today known as plasma membrane. Today it is known as plasma membrane. He also concluded that based on his studies on plant tissue that they told that presence of cell wall is a unique character of the plant cells. It is a unique character of plant cell. Okay, and on the basis of this, Swan proposed a hypothesis that the bodies of animals and plants are composed of cells and product of slabs. So who proposed that it is made up of composed of cells and products of cells? Swan. But Sladen and Swan combinedly given formulated a cell theory. So next we talk about cell theory, that is formulated by Sladen and Swan, and they put some of the point in which number one, that each cell is made up of protoplasm having nucleus and bounded by a cell membrane. So there is a presence of nucleus and cell membrane. Second, organisms are composed of composed of cell and their products. And third point, that the functions of an organism is an outcome of activities and interaction of its constituent cells. So these are the points which was given by Slater and Swan during this, but they did not explain how new cells form. He was not able to explain that how the new cells were formed. So later on this cell theory was modified So cell theory given by Sladen and Swan, that is modified by Rudolf Virchow, so this is the fact based question you remember, and they asked directly question from that, and he explained that cells develop from pre-existing cells. Cells develop or arise from pre-existing cells, that is omnicellular acellular, and it is also known as law of cell lineage. It is also known as law of cell lineage. Number of other modifications were coined out in theory. The modern cell theory is also known as cell principle or cell doctrine. So we remember modern cell theory known as cell principle or cell doctrine. Okay, so that is about what a cell theory. Now there are some exceptions to the cell theory as well, or we can tell them drawbacks of the cell theory or exceptions of the cell theory, that also we take into consideration. So exception to cell theory, or which cell theory is not applied are: first, viruses, they cannot be explained using this cell theory. Second, bacteria and blue-green algae do not have organized nucleus. Third point, certain fungi like Rhizopus have hyphae composed of multinucleated mass of cytoplasm. So Rhizopus have a cenocyte. Okay. Acetabularia is a fourth example, that is a green algae which has a uninucleated differentiated body. Uninucleated differentiated body means they are acellular. Next to that, sieve tube and mature RBC lac nuclei. Okay, so these are the some of the exception over which the cell theory cannot be applied. So we remember this exception as well, that they are not a part of cell theory. Now we talk about some of the points regarding the overview of cell. Now when we note down some of the points with respect to overview of cell, we find that cells differ greatly in size, shape and activities. Cells differ greatly in size, shape and activities. For example, if you see, then mycoplasma is the smallest cell and it has only 0.3 micrometer in length. It is 0.3 micrometer in length. While when we talk about bacteria, mycoplasma is also a bacteria which is wall-less. Now if we talk about the size of bacteria, the size varies from 3 to 5 micrometer. The largest isolated single cell is egg of ostrich. Among multicellular organism, human RBC are 7 micrometer in size. Nerve cell are longest cell, some of the longest cells. So cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function they perform. So one of the important point there you should remember that the shape of cell may vary with the function they perform. Okay, so this is about the overview. Now after that we are going to talk about in detail a prokaryotic cell and eukaryotic cell. Okay, so let's start with first our prokaryotic cell. So we start with prokaryotic cell. Now prokaryotic cell, if you talk about a structure, this include bacteria. Okay, so if we draw the structure of this prokaryotic cell, we have to remember that these are the cells without true nucleus and membrane bound organelles are absent. So we remember membrane bound organelles are absent. It does not mean organelles are absent. Organelles are present but they are membrane less, like the ribosome. They can be present in case of prokaryotic cell. Now first we should understand the structure. If we understand the structure in detail, then we are able to identify all the components of this part. Okay, so when we see the structure somewhat look like this. This is a plasma membrane. Outer to plasma membrane we find a cell wall. and outer to cell wall you find a glycocalyx. Okay, so this is the glycocalyx. Then inner to that there is a presence of cell wall, and inner to cell wall there is a plasma membrane, and these three combinedly called as cell envelope. Okay, next inner to here also you can see some extension like this, and we call them flagella. Some extension like this, they are known as pilus, and plural is pili. Okay, they have respiratory enzymes associated with the plasma membrane. Centrally they have a nucleoid. True nucleus is absent, so they have a nucleoid. In spite of nucleoid there is a presence of another structure like circular DNA. This is known as plasmid. This is known as nucleoid. Then you find some structure like this that are named as polysome. Okay, they have some of the enzymes, some of the proteins, and all these are present. One more thing there I find, that is a presence of inclusion bodies. Now polysome is found, but with that 70 as ribosome is found free in this cytoplasm. Okay, so this type of structure is visible in case of this one envelope system, then pili, flagella, nucleoid, polysome, respiratory enzymes. Okay, all this with inclusion bodies. One more thing, you will find some of the fimbriae like structure like this. These are known as fimbriae because cilia is absent in case of prokaryote. They have a fimbriae, bristol like structures are present. So this is the structure of actually a prokaryotic cell. Now we need to talk about what particularly each and every part in detail. Be remember that membrane bound organelle are absent in prokaryotic cell. So first in prokaryotic cell we talk about cell envelope. Now this cell envelope is single envelope system in case of prokaryote, but similarly eukaryote have double envelope system, means they have a membrane bound organelle which form another envelope. Outermost is the single envelope. Now this envelope or cell envelope consists of or made up of first glycocalyx, and glycocalyx is the outermost layer. Now this glycocalyx is of two types, that is capsule or slime layer. Capsule is give shiny colonies, whereas slime layer give rough colonies. Capsule has virulent nature and this has a non-virulent nature. Now this capsule are hard or tough, because of this they gives a particular colony, but they are rough, so they gives a rough colony. So this is the characteristic of what glycocalyx. Now this capsule help bacteria to hide against host immune system. But this slime layer help in attachment of bacteria to the another bacteria. Okay, so this is about the what glycocalyx. Next, inner to that, we talk about second part, that is cell wall, and cell wall is the middle layer. Now cell wall in prokaryote, it is made up of peptidoglycan or murine or mucopeptide. Means peptidoglycan is also known as murine or mucopeptide, and it is a polymer of NAG, that is N-acetyl glucosamine, and NAM, that is a N-acetyl muramic acid. So they are the polymer of NAG, N-acetyl glucosamine, and N-acetyl muramic acid. Okay, and if you see the structure of this, you find NAG, NAM, NAG, like this we find a chains. And these chains are joined by tetrapeptide. These chains are joined by tetrapeptide. It is a tetrapeptide is made up of 4 amino acids. Like this, there is a formation of this peptidoglycan in the cell wall. Third is plasma membrane. Now plasma membrane is also known as cell membrane or plasma lemma. It is the innermost layer of cell envelope. Now this plasma membrane has a phospholipid bilayer structure. Now this phospholipid are arranged somewhat like this. So there is a particularly these are the intrinsic protein, there is some extrinsic protein, and they have this structure that is of phospholipid. Now in this phospholipid, if you see the structure, you will find like this. This is a head and this is a tail part. This head are hydrophilic and this tail part are hydrophobic. Now in the plasma membrane of prokaryote, cholesterol are absent and hopanoids like lipids are present, which provide them a stability. So this is the structure of plasma membrane in case of the prokaryote. Now next to cell envelope, we talk about nucleoid. Now nucleoid is also known as false nucleus means true nucleus is absent. Now this nucleoid is made up of double stranded circular DNA and non-histone protein like polyamines. Okay, so this polyamines and double stranded circular DNA combinedly form a nucleoid. This nucleoid is also known as incipient nucleus or genophore or chromonyme or prochromosome or single chromosome. These are the names of the particular subcomponent in case of nucleoid. So nucleoid in this nuclear envelope is absent. Next to that, we talk about a third structure, that is plasmid. Now plasmid is defined as extra chromosomal self replicating double stranded DNA, as we have seen in case of the diagram. They have defined as extra chromosomal self replicating double stranded DNA, and this plasmid are of two types, that is F plasmid and R plasmid. Now F plasmid is known as fertility plasmid, which help in conjugation, and this R plasmid is known as resistance plasmid, which provide resistance against antibiotic. They provide a resistance against antibiotic. So this is a plasmid. So we remember to define this particular definition of plasmid, because about the definition, from this definition only 2 times question asked in case of NEET. So we remember that what is plasmid? It is the extra chromosomal double stranded circularity self replicating DNA. Extra chromosome. Why it is named as? Because it is extra to the nucleoid, and nucleoid is also known as single chromosome, as we have seen. So it is extra to the nucleoid, because of this it is known as extra chromosome. They can replicate itself, and it is made up of double stranded DNA. Next we talk about a structure that is known as polysome or polyribosome or ergasome. Now polysome, in polysome, or this what happens, there is a one mRNA or messenger RNA, and over which when several ribosomes are attached, then it is known as polyribosome. So polyribosome consists of many ribosomes on a single mRNA. Now this ribosome are of 70S type, and 70S ribosome have two things, that is large subunit and small subunit. Large subunit is 50S and small subunit is 30S. So that is a ribosome 70S type present on the mRNA, many ribosomes, that is known as polysome or polyribosome. This is also important question, because they ask usually in NEET. Okay, after polysome we talk about next structure, that is inclusion bodies. Now these inclusion bodies are the food storing bodies in case of the prokaryotes. So these are food storing bodies which are present in cytoplasm, not bounded by they are not bounded by any membrane. Not bounded by any membrane system. Okay, they lie free in the cytoplasm. If we talk about example, then example is the phosphate granule, synophysin granule, and glycogen granule. Then one more is gas vacuole, and this gas vacuole are found in blue green algae and purple and green photosynthetic bacteria. Okay, in this they are found. Now just we have discussed about the ribosome. The size of ribosome is 15 nanometer by 20 nanometer in size. Be remember the size of this particular ribosome. And what is the function of the ribosome? Ribosome play role in synthesis of protein. They play role in synthesis of protein. Next is mesosome. Mesosome are invagination of plasma membrane. Invagination of plasma membrane means, suppose this is a plasma membrane, this is a cell wall, and this is a plasma membrane, then here you will find this invagination, and this is known as mesosome. This mesosome play role in cell wall formation, DNA replication, and distribution of daughter cells. Distribution of daughter cells. And they increase surface area of plasma membrane. Surface area of plasma membrane. In some prokaryotes we even find the structure that is known as chromatophores. So this chromatophores are membranous extension into cytoplasm and contain pigment. They are found in cyanobacteria, means blue-green algae. Next we talk about structure of flagella. Now when we see the structure of flagella, they have some ring in a basal body. Like this flagella is visible. This is cell wall and this is plasma membrane. They have this ring, they are named as LPSM ring. And this is hook and this is filament. Now filament is made up of flagellin protein. We remember in case of prokaryote, this filament is made up of flagellin protein. This LPSM ring combinedly form known as basal body. So basal body, hook and filament combinedly form a flagella. If we talk about function of this flagella, it provides a motility or movement to the particular cell. Next to that we talk about another structure, that is a pilus or pili. This pilus or pili is made up of pillin protein. Suppose this is the bacteria. This bacteria have this nucleoid and it have another plasmid. If suppose this is a F plasmid, then it has a gene that is known as F factor. Now this gene produce F factor or gene produce a pillin protein, and that pillin protein accumulate and form a structure that is known as pilus. This pilus play a role in conjugation, as we have discussed in case of a biological classification. Next to pilus we have some structure that are named as fimbriae. Fimbriae are bristle like structures growing from the cell wall, and they help bacteria for attachment to rock. They help in attachment of the rock. So when bacteria is moving through any stream and it thinks that bacteria now want to attach and take rest, then this time they use this fimbriae and get attached to the bacteria. Next we talk about shape of bacteria or prokaryotic cell. There are four main shape of this bacteria or a prokaryotic cell. Okay, number one, coccus. Coccus are spherical or oval. The plural is cocci. Okay, and they are non-motile. Okay, because absent. Second is bacillus, plural is bacilli. They are rod shaped. Okay, and they motile or non-motile, means flagella may be present, maybe not present. Then third type is vibrio, that is comma shaped. They are comma shaped, and in this they appear somewhat like this, and in this also they have their flagella, so they are also motile. And fourth type, that is spirillum. And spirillum, you find a group of flagella at the both, and so this is also motile, and it look like half screw shape. Okay, and it is motile. Now out of this you have to remember one important point is that this bacillus is the most common shape of bacteria. Okay, it is the most common shape of bacteria. Now after studying the structure of prokaryotic cell and their shape, next we talk about one staining procedure which occur in case of bacteria, that is known as gram staining. And based on gram staining, that two type of bacteria are derived, that is a gram positive and gram negative. This gram staining developed by Christian Gram, and based on the name of the scientist it is known as gram staining. Now what they did, they have taken one bacterial culture in a test tube. This is a test tube. They have taken one bacterial culture. Now from this they have taken one loop full of bacteria and kept in on a slide. So on a slide, if we take a loop full of bacteria and rotate it, it will create bacterial smear. Then sometimes we heat fix also to fix it properly. Then we stain with gram stain or Gentian violet. Now this Gentian violet is purple color stain, so it gives purple color. Now after Gentian violet staining, iodine staining as a mordant, means it press it. After that we give a water wash. So in this water wash what will happen, the extra stain get removed. So now this particular bacteria have a purple color. So then we carry out alcohol wash or ethyl alcohol wash. So after alcohol wash, what will happen, some bacteria retain this purple color and some bacteria will lose it. Some bacteria retain purple color. These are known as gram positive. But some bacteria lose this stain, and they lose stain, so they become colorless. So they are again counter stain by safranin, and when counter staining occur by safranin, they give a pink colored colonies, and this pink colored colonies bacteria are referred as gram negative bacteria because they have removed this particular junction violet stain. So now the question is, why gram positive bacteria retain this particular stain and why gram negative bacteria lose this? So for that we have to study the structure of their cell wall. So cell wall of gram positive and gram negative bacteria. In gram positive it is single layered, whereas in gram negative it is bilayered. So if you see in case of gram positive, it look like this, and this is made up of peptidoglycan. Now in this it is bilayered, they have this inner layer and one is outer layer. So this outer layer is known as lipopolysaccharide or LPS layer, and it is rich in lipid, whereas this is the inner wall which is made up of peptidoglycan. So now what happens when these both bacteria are stained with the gentian violet first, this bacteria receive this gentian violet. Now they take up this whole wall take up this gentian violet, but when gentian violet or purple stain, we stain this gram negative bacteria, this purple stain come in which part? LPS layer. So when later on we treat with, we have first initiated with the gentian violet, the purplish color is given to this outer wall. But when we treat with the alcohol will, or ethyl alcohol will break, or acetone will break this lipid rich layer, that is a lipopolysaccharide layer. So this stain will also get removed and this bacteria become what? Colorless. So again we counter stain with the safranin, and the safranin come and bind with this particular layer, so it give a pink color to the gram negative bacteria. So these are the difference. Because of this, gram positive bacteria are purple colored, whereas gram negative bacteria become pink colored due to the safranin staining. So this is all about the prokaryotic cell, which is necessary with respect to cell structure that we have studied. Thank you.